Polygenic inheritance results when you have more than one gene contributing to the expression of a particular trait. We generally see these traits exhibited as a continuum of phenotypes. Two well-known examples are human skin color and human height. There are at least 3 genes at different chromosomal locations responsible for skin color. This leads to a wider variation of phenotypes along a bell curve distribution. There is a wide variation between extreme phenotypes, with most individuals having intermediate phenotypes. In skin color, the dominant genes are additive, with each dominant gene adding 1 unit of color to the genotype. With more dominant genes, the phenotype, which is the appearance, gets darker. Multiple alleles are when 3 or more alleles control a trait. The best known human example of multiple alleles is the ABO blood types. There are possible alleles here: IA, which codes for the A glycoprotein, IB, which codes for the B glycoprotein, and I, which codes for neither. This results in 6 possible genotypes and 4 phenotypes. A and B are codominant to each other and dominant over I. Codominance is similar to incomplete dominance, but a little bit different, because in this case, the heterozygous individual expresses both homozygous conditions simultaneously. In incomplete dominance, a red allele and a white allele are expressed as pink. In codominance, a red allele and a white allele could be expressed as a red and white stripe. Both alleles would show up. An example of this is the AB and AB blood types in humans. If you have an A allele, then you produce A glycoproteins on the surface of their red blood cell. If they have a B allele, then you'll produce B glycoproteins on the surface. But if you receive an A and a B allele, you will produce both A and B glycoproteins. This Punnett square shows how the A glycoproteins, B glycoproteins, and the AB glycoproteins that are together are shown in a Punnett square. Incomplete dominance is when the heterozygous individual exhibits a third phenotype that is in between the 2 homozygous phenotypes. An example is the color of flowers in snapdragons. There would be a red allele and a white allele. Flowers that have 2 red alleles are red. Flowers that have 2 white alleles are white. But if you have 1 red and 1 white, red does not dominate, it's pink. Incomplete dominance alters the phenotype ratio we see because now the phenotype ratio is equal to the genotype ratio. This Punnett square shows a homozygous red flower with a homozygous white flower, which produces heterozygous pink flowers. Epistasis is when 2 or more genes control the expression of a single trait. One example of this is the inheritance of coat color in Labrador retrievers. This involves the interaction of 2 different genes. One gene, we'll call it big E, determines whether or not pigment will be produced. If a dog inherits at least 1 dominant allele, then this allows the gene for pigment to be expressed. The big B allele is dominant to the little b allele. Dogs with 1 or more big E's inheriting 1 or 2 big B's alleles are black, while dogs with 1 or more big E alleles inheriting 2 little b alleles are brown. If a dog inherits 2 little E's, he will be white, regardless of which combination of B alleles he inherits. Mendel's genetics. Mendel was an Austrian monk who was studying pea plants. During his investigations, he discovered the law of dominance, the law of segregation, and the law of independent assortment. However, very few traits are expressed in simple dominant recessive patterns. So knowing the extensions to genetics allows you to understand why different genotypes express different phenotypes.